So hopefully that's loaded up for, okay. So if you're still there, hit terminal, and that should take you to the home directory that we're going to be working out of. Thanks to Mike Lee for setting this up for me, actually. Okay, so in the GitHub repo to which I sent out the link, there's a walkthrough, and I'm basically going to follow the commands in this walkthrough to sort of demonstrate how you can use SortMeRNA. So first things first, we're going to hit ls, and we're going to see what's around us. Okay, so I like to do ls-lht because it's easier for me to read this kind of output. Okay, so you have your basically two folders that are important. You have your reads, you have your rRNA databases. So just quick note of warning is that, well, let's take a look at the rRNA databases. So these actually come with the SortMeRNA package. So this is what they look like. So two files are the key ones. And this is just two of the databases. And it's a small subset of the databases. And the reason for that is just for tutorial purposes, it's going to run a lot faster. And I think that there is a 100 megabyte file limit for uploading stuff into this binder. So I only uploaded two databases, and that's the 16S and the 23S for bacteria. Another thing that will not be covered in this tutorial is the indexing of the databases, but that's a fairly straightforward command. If you go off of the SortMeRNA manual, you basically run a command to generate these indexed versions of the database. But I decided not to include that as part of the tutorial for some reason. Don't remember why, but no use in talking about it now. All right, so let's take a look at one of these files. So we're going to less into the 16S bacterial database and just see what it looks like. That's not, okay, I did that wrong. Okay, so basically you have your 16S sequences here. Pretty simple, straightforward. I assume most of you have seen a FASTA file. So it's basically a FASTA file, and it's used as a reference database for your reads. So we're going to go back up to directories, and we're going to take a look into the reads folder. So the reads folder has 3 files in it. And for this tutorial, I included a set of sequences that are paired end sequencing. So you have your forward reads and you have reverse reads. And there's also a set of sequences that I got back from a single end run. So let's just look and see what a FASTQ file looks like. Looks good. And these have all been QC'd already. So these are all trimmed versions of the files. Okay. So just for fun, I actually thought to include Flash into this binder, just to kind of see how that runs. So Flash is a very easy program to run, but if you do Flash-H, you're actually going to get a fairly long help menu, and then there's a lot of different things that you could change. I usually just run it using the defaults and just see what the output looks like. So, and it's really fast, and especially with the fact that if you look at the file sizes, these have all been subsampled. So you know, hopefully when you get back your files from the sequencer, they're not going to be 7 megabytes in size, because that's tiny. You know, you're only looking at you know, this divided by 4 number of reads. So it's really not a lot of reads, but so Flash is going to run super fast. So you just run Flash, forward, and then reverse, and then you hit enter. and then it, boom, less than a second, it's done. And then since I didn't provide with Flash with the -O flag, it's gonna make the it's gonna give you the default output file name. That's gonna be out dot. So the not combined 1 are the forward reads that didn't combine, and the out combined 2 are the reverse reads that didn't combine, and the out dot extended frags are the reads that combined. Right, so you see that most of the reads combined, and then if you look at the output, you'll see that 63% of the reads combined. I'm not sure what the reason for that is. So if you play around with the options and you want to improve, and you want to see if more reads will combine, if, for example, you allow outies, so reads by default will be tried to combine by Flash using the any orientation. So like this, we can also allow outies. You can also play around with the minimum overlap and the maximum overlap for reads. So I think for the minimum overlap, it requires that there's at least 10 base pairs of overlap between the forward and reverse reads to combine them. You can lower this to 5 and see if that's going to improve. You can also increase the max overlap. But yeah. So actually, let's move on with the SortMeRNA because that's what the tutorial is. So we're gonna print the help menu for SortMeRNA and see what that looks like. It's pretty long. So let's start breaking this down. So I'm actually gonna start with a single end reads. I'm gonna start with this. Oops. I'm gonna start with the single end MBIC. So let's see. This is what the command would look like. SortMeRNA. So -- reads. Pretty simple. There's only 2 required arguments actually, but we're gonna play around with more of them. So -- reads, and then we're gonna give it the reads file. And we're gonna wanna give it the -- reference file. Okay. So this is going to be the one of the FASTA files, and we're gonna provide the 16S first. So if you actually look in the, I'm gonna copy this. I'm just gonna list the files in the rRNA databases. Okay, so when you provide the ref, you want to provide the FASTA file, and you want these files, the .dat, the .birthstreet, .dat. You want these files to be in the same directory as this FASTA file because SortMeRNA is going to look for those files in the same directory, but you want to omit those from the arguments. So when you do -- ref, you just want to provide this FASTA file. And then we're going to do the same thing for the 23S database. All right, there you go. So as you can see, we're providing the -- ref argument twice. All right, so let's see what else there is. So we're going to want to include the -- fastx flag because we want the output to be in fastq format. And then we're going to want to provide the -- aligned. What is that? -- aligned flag. And that's going to output in fastq format the reads that align to the database. So therefore, these reads are going to be your rRNA reads. Right? So we're gonna name this file the same as the original file, except we're gonna add a little note here that these are rRNA reads. And then we're going to add the -- other. And -- other means that these reads did not align to any of the databases. And so these are going to be your mRNA reads. Okay. I think for simplicity purposes, we're gonna call that, and we're gonna, yeah, I think that's gonna be it for this round. So we're gonna hit enter, and it's going to run, and it should finish in less than a minute because there's not a lot of reads for it to go through. If you want it to run faster, there's a -- threads, but I'm really not sure how this works in Jupyter Binder, so we're gonna use the default 1 number of cores for this, so we don't break anything.
I think the binder by definition only has 1 thread.
Okay, that's good to know. What happens if you do -t like 16? I think it's just not. It's just gonna use 1 still.
Well, can you repeat the question?
If you do like -- threads 16, if you try to run something with 16 threads, is that going to break anything, or is it just going to use the 1 that's provided with?
I don't know, but when I was recording another tutorial that uses all the threads available, it was just using the 1. So that's how I figured it out.
It's good to know. All right, thanks, Ella. All right, so it's done. It's finished. So let's do ls-lht to see what we have. All right, so you can see that there's 3 new files that are created in our directory. There's the mRNA, and there's the rRNA. So we could see that it's actually, this sample was mostly rRNA, and a small proportion of the reads are mRNA. So not good, but that's okay. It's not the end of the world, but let's take a look at the log file and see what the log file looks like. Okay, so from this we could see that 93.52% of the reads passed the e-value threshold. So that's another thing that I actually didn't really discuss, is that you could play around with the e-value for this, and you could lower the e-value if you want SortMeRNA to be more stringent in what it calls rRNA. I usually go with the default e-value of 10 to the minus 10. I have to look into that. It's 10 to the minus 5 or 10 to the minus 10. But you can see that a big proportion of our reads, so it almost seems like the sample was not rRNA depleted, and it's just total RNA. So we have 93.52% of rRNA, and 6.48% of the reads are mRNA. So out of those that did pass the e-value threshold and are therefore labeled rRNA, 68.16% are 23S, and 25.35% of the RNA is 16S. Okay. And I think that's pretty much the main takeaway from this log file. Okay, so let's do one more quick SortMeRNA run, and let's do this on the forward and reverse. So let's say you have paired-end sequencing, and you want to run SortMeRNA on that. So it's very simple. It's actually almost as simple as what we just ran. But let me just copy and paste the commands from my walkthrough. So SortMeRNA -- reads forward -- reads reverse. So the order is actually pretty important here. So if you do -- reads, the first file that you provide should be your forward reads, and then the second -- reads file you provide should be your reverse reads. Okay, let's provide the reference databases right now. Let's give us the 23S and the 16S. Okay. We're going to do the -- fastx. So the output is in FASTQ format, so we can use it later on. Dash dash other. So this is going to be our mRNA output. So let me actually print the menu again, because there's a couple of things in there that I want to discuss. Okay. So if you're doing forward and reverse reads, if you're providing paired and reads to SortMeRNA, you're going to want to add this dash dash out to option. And what that's going to do is that's going to output paired reads into separate files. So you're going to basically retain the same organization with the forward as forward reads and reverse as reverse reads, and they're going to be paired, right? So whoops. So that means that if you are doing anything downstream that requires you to provide forward and reverse reads as paired files, that's not going to mess with it. So the fidelity of the reads is going to be retained in those two files. Another important thing that you need to consider are these two options, the dash dash paired in and the dash dash paired out. So, and this is sort of a judgment call. So if one of the paired in reads is aligned and the other one doesn't, do you want to keep it? Do you want to keep both reads in, or do you want to throw both of the reads out? So I generally go with paired out. So if you have paired reads and one of the reads appears to be mapping significantly to an rRNA database, but the other one doesn't, you would throw both of the reads out. So we're going to go ahead and we're going to add this flag. Okay, and that's pretty much it. So then you hit enter. Whoops. I'm not sure what this error is, but I've had this error quite a few times here, and I don't get this error if I run this program on my Mac or Jupyter or a regular server that I work out of. So it seems to be a binder specific error, but basically you just get rid of this directory. It needs to be empty to rerun SortMeRNA. Okay, so we're going to hit enter. And we're going to wait for it to run. So that's pretty much it. There's one more command that I do want to go over, and that's sort of this, that's one that takes advantage of this OTU picking capability of SortMeRNA, which I think is pretty cool. I've never actually, I mean, I usually include it in my commands, but I've never used these OTU tables that SortMeRNA outputs. But I think it's pretty cool that it has this capability, so I kind of want to showcase it. All right, so finished. Okay, let's see what files it created. So created 2 files. Yeah, so there you go. So you have your forward reads. This is your 1P, forward reads, mRNA, and then you have your reverse reads. And as we could see, most of the reads actually are mRNA. So the sample is actually ribo-depleted successfully. Yeah, so let's do this OTU picking command. And for this, I'm going to provide the single end file. And we're going to provide the databases. So keep in mind that the more databases you provide, the more RNA is going to be depleted, depending on where your sample is from. But in my case, if I have samples with eukaryotic and bacterial DNA, so if I provided just the bacterial 16S and 23S databases, I will have a certain proportion of my reads removed as rRNA, but that's not gonna remove the eukaryotic rRNA because those aren't gonna match the databases. So the more databases you provide, the higher likelihood it is that you're gonna be left with less sequences to play around with, but they're gonna be mRNA for sure. All right, so this looks like exactly like the command I ran before, but we're gonna add the -- OTU map as well. I'm gonna move that. So once it finishes, it'll output the same files that it outputted before, except there'll be an additional file that will be basically a counts table. And this counts table is based off of the read similarity of the reads that map to the 16S database. So it's going to cluster those reads into OTUs essentially, and the counts are going to be based on the read abundance. Okay, so these are the two files that we got before. This is the log, and this is the OTU table. Let's look at this. Oh, that's huge. Okay, let's do the less dash capital S. Okay, so this is, yeah. So basically, each of these, so the first column is the sequence to which a read map to. So this is a sequence that is derived from the database, essentially. And each of these columns following the sequence to the tab delimited list of headers. Each of these is a header from your FASTQ file. So each of these is a read that mapped to a particular FASTA sequence in your database, right? So if you're really
this is basically a closed reference OTU clustering?
Yeah, that's what it seems like. But I thought I gave you a table. Maybe they changed it, or maybe I'm misremembering, but it seems like you have to do this yourself. Like if you want to count how many reads map to each sequence.
And maybe I missed it, but what's the percent ID of OTU clustering here?
So there's no clustering actually. It's just, well, so the databases themselves are clustered, and I'm not sure. I think that it's either 95% or 97%. So the databases themselves are clustered 16S sequences. So each sequence, or the 23S sequences in the database, is a representative of a set of sequences. And this mapping is e-value based. So this is a local alignment to a sequence in the database. And if it passes the e-value threshold, then it's assigned to this row. Does that make sense?
Yep, gotcha.
And I think that with SortMeRNA, as far as I remember, you have more or less control. I think that, they changed two versions over the last relatively short period of time. So there's been significant changes that I haven't kept up with, but there was a time where you could decide whether you want a rRNA database, like a 16S rRNA database that is really big, that's clustered at the 97% level, or if you want a more broader database with less sequences that are clustered at a 90% identity level. And that will determine how fast SortMeRNA runs. So if you want it to run really fast and you want to give it a smaller database, or if time is not an issue, then you want to be as comprehensive as you can. So you can provide it with the entirety of Silva if you want. If you want to download the entire, I think it's a gigabyte, that whole database. You download it, you can make a SortMeRNA index out of that, and then you could use that as a reference pull out RNA reads.